This protocol is used to quantitatively study the assembly and structure of the axon initial segments of hippocampal neurons that lack pre-assembled AIS due to the absence of a giant ankyrin-G. To begin, dissect 6 to 8 hippocampi from postnatal one-day-old pups with HBSS medium in a petri dish. Chop the hippocampi with dissection scissors into smaller pieces and transfer them from the dish to a 15 milliliter tube. Wash the hippocampi twice with 5 milliliters of HBSS and leave them in 4.5 milliliters of HBSS after the wash. Add 0.5 milliliters of 2.5% trypsin into 4.5 milliliters of HBSS and incubate in a 37 degrees Celsius water bath for 15 minutes, inverting the tube every 5 minutes. Well-digested hippocampi should become sticky and form a cluster. If needed, extend the digestion for 5 more minutes. Wash hippocampi with HBSS 3 times for 5 minutes per wash. After the wash, add 2 milliliters of HBSS and pipette the hippocampi up and down with a Pasteur pipette 15 times. Triturate the tissue with a fire-polished Pasteur pipette 10 times and rest the tube for 5 minutes until all chunks set to the bottom. Repeat the trituration with the remaining chunks until most of them have disappeared. Then, use a 1 milliliter pipette tip to gently transfer the supernatant containing the dissociated neurons to plating dishes. Add it directly to the